Okay, we are moving right along to gluconeogenesis. We finished glycolysis and we ended at pyruvate. So gluconeogenesis is starting from pyruvate and it's going back up to glucose. So it is the exact opposite of glycolysis. And a lot of the steps are the exact same. There are 3 steps that are different that use different enzymes. So gluconeogenesis would happen if you have low blood sugar and you need to create more glucose. So let's start with right where we left off with pyruvate. Now, the first step to go from pyruvate back to PEP is going to have an intermediate. So this is the only step that's going to have an extra intermediate. So this extra intermediate is called oxaloacetate. And this reaction is catalyzed by an enzyme called pyruvate carboxylase. And this step as well uses an ATP and turns it into an ATP because it needs a phosphate. So now we can go back up to PEP. We are going to lose an inorganic phosphate, the one that we gained from down here from this ATP. We're going to go ahead and just lose it straight away. And instead, we're going to pick up a phosphate from GTP. So not ATP, GTP, and it's going to turn it into GDP. And this reaction is catalyzed by an enzyme called phosphoenolpyruvate carboxykinase. That is a mouthful, but you can just call it PEPCK. So just to get back from pyruvate to PEP, we did this whole loop around thing. And oxaloacetate, as you will learn later, is very important, but for now it is just an intermediate in that step. And so from here, actually the next few steps in gluconeogenesis is the exact same as glycolysis, just in reverse. So this comes all the way back up, and just to note, we'll put our arrows this way to show that it can go either way. So all of these reactions can happen in reverse. Step 1 and step 3 are going to have different enzymes because it is using ATP to catalyze that reaction. These are irreversible reactions. A lot of times we would like to call step 3 the committed step, so we can go ahead and put that here. The committed step, because once you go through step 3, like, you're kind of stuck doing glycolysis all the way down. So we need a way to work around PFK in step 3 to get back from fructose 6-bisphosphate to fructose 6-phosphate. Let's draw our arrows. Instead, we are going to use an enzyme called fructose 1,6-bisphosphate. Bisphosphate. Okay, and this is also a control point. Okay, so we got past step 3. Step 2 is reversible. We can just use the same enzyme here. Now step 1, again, it uses ATP, so we're going to have to find a separate way around to get back to glucose. So instead of hexokinase, we're going to use glucose 6-phosphate. Now, these enzyme names can be semi-easy to remember just because they add taste to the end of the reagent. So fructose 6-bisphosphate goes, the enzyme is fructose 1,6-bisphosphate, and then glucose 6-phosphate, the enzyme is glucose 6-phosphatase. Whoops, I spelled that wrong. Phosphatase. So that is gluconeogenesis. Really the only difference is 3 steps, and the 3 steps that are irreversible that you need a separate enzyme to reverse is step 10, step 3, and step 1. And so those are the key regulatory steps. That's why we need different enzymes. I'll go ahead and label Genesis. Gluconeogenesis is pyruvate to glucose. Okay, awesome. That's really all there is for gluconeogenesis. Again, all of the notes that I filled out and went through, I will put in the notes page of the link tree that is in my bio. This will be under biochemistry, and then the PDF will be gluconeogenesis.